Hello everybody, welcome to NEET Crash Course Biology. Welcome to Deeksha Online. So now we are going to do a quick review of a quick synopsis of cell, the unit of life. Now you all are aware that the term cell was coined by Robert Hooke, and Anton von Leeuwenhoek with his earliest microscope was the first one who saw different cells under the microscope. And you know that organisms may be unicellular or single celled, and there may be multicellular organisms. So ultimate thing is the cell is the basic unit of life. The cell is the most fundamental structural and functional unit of life. It was Matthias Kleeden in 1838 who was a German botanist, and Theodor Schwann who was a German physician. However, it's wrongly mentioned as a British zoologist in the NCERT. So remember, Theodor Schwann in 1839 who was a German physician, and Matthias Kleeden in 1838 who was a German botanist, collectively put forth the cell theory. Matthias Kleeden was exclusively learning on plant tissues, and he was studying on plant tissues, and basically Theodor Schwann was the first one who proposed or who reported that cells have an outermost thin membrane that is called plasma membrane. So both of them studied independently, and they identified that basically tissues are made up of cells. But however, the Schleiden and Schwann's theory did not explain the origin of cells, for which Rudolf Virchow put forth the theory, that is the theory of continuation of life. That is basically how did the cell come into existence. He said that new cells develop by cell division from already existing or pre-existing cells, that is omnis cellula e cellula. So Rudolf Virchow in the year 1855 proposed that a cell doesn't spontaneously or come into existence as it is, but for a cell to exist there should be a pre-existing cell. And how does it come forth? By the process of cell division. And basically Schleiden and Schwann's theory was put together, and it was called the cell theory, according to which all living organisms are composed of cells and products of cells, and all cells arise from pre-existing cells. When the cell was observed under the electron microscope in the 1950s, of course, the electron microscope came into existence. They came to be identified as prokaryotic cells and eukaryotic cells. Let us take a simple diagram for a prokaryotic cell, a bacterial cell. The most common example for a prokaryotic cell is a bacterial cell. Notice that a bacterial cell has a cell wall on its outside, and it has a plasma membrane on the inside. So the outer layer which it is covered with is referred to as the cell wall. Outer to the cell wall you can see a slimy covering which is labeled here as the mucilage, and the innermost layer is what is referred to as the plasma membrane. Now I would like to draw your attention onto these foldings that you can see in the membrane. These inner foldings which are found in the membrane are referred to as the mesosomes. The mesosomes help in cell wall synthesis, they help in replication and distribution of the DNA between the daughter cells, they help in secretion. They also contain photosynthetic pigments in the case of photosynthetic bacteria. They may contain respiratory pigments. They contain a lot of enzymes. They increase the surface area for enzyme action. And notice how the DNA of the bacterial cell is. It is a prokaryotic cell. It has a naked double-stranded DNA. And this naked double-stranded DNA is called the nucleoid. You cannot see a nuclear membrane unlike in the case of a eukaryotic cell. Okay, and then can you see these small units called ribosomes and how they are connected to each other to form a beaded chain like structure which is referred to as a polysome. So you can identify that they have polysomes in their cell. They also have ergastic substances or inclusion bodies. Inclusion bodies are storage substances, like you can see here there is a glycogen particle that is there, glycogen droplets are there. Some forms exhibit a gas vacuole. A gas vacuole is not covered with a membrane, or it may be covered with a non-unit membrane, and these gas vacuoles usually help in providing buoyancy. Then you have protein particles. Along with these glycogen particles you may also find in some of the bacteria you may find phosphate granules. And if it is a bacteria which is a cyanobacteria, then you can find cyanophycean granules, which is basically the cyanophycean starch. So basically a lot of inclusion bodies can be found, and generally glycogen and phosphate and synophysin granules are not bound with the membrane. Sometimes you may find a gas vacuole. Along with gas vacuole, you may also find certain sulfur granules that may be present, or you may find pH, that is PBHP, polybeta hydroxybutyrate crystals, which are covered by a non-unit membrane. A non-unit membrane means, say for example, this is the gas that is accumulated in the center. It may have a single layer of lipids, not a bilipid layer that we see in a typical plasma membrane, which is called the unit membrane. So there may be a covering of, that each lipid molecule has a hydrophilic head and a hydrophobic tail. This kind of covering can be considered to be a non-unit membrane. And in a typical prokaryotic cell, you will not find any membrane bound cell organelles, except you will only find ribosomes, and that is basically ribosomes are common to both prokaryotic and eukaryotic. Otherwise, you will not find any endoplasmic reticulum, no nuclear membrane, no Golgi apparatus, no lysosome, no peroxisomes, no glyoxysomes. So you will not find any membrane bound structures. Notice that there are locomotory structures which are called as flagellae. The flagellum of bacterial cell has a basal body attached partly to the plasma membrane on the inside and the cell wall on the outside, and it has a curved hook like this, and from the curved hook a solid, a rigid or a hollow structure originates, and it is made up of individual units or protein subunits which are referred to as flagellin, unlike the flagellar structure that you see in eukaryotic cells. Then you see certain bristles which are projecting out of the cell, which help in anchorage or attaching to certain substratum that is referred to as fimbria. Fimbria is also a solid structure which is said to be made up of 3 to 10 nanometer sized protein particles, or basically the protein units which make up the fimbria. Sometimes on a bacterial cell you will find these hollow tube-like structures. Remember that these hollow tube-like structures which help in transferring the genetic material, that is conjugation between the two bacterial cells, this is referred to as a pilus. It is identified that pilus is only seen in gram-negative forms of bacteria. Sometimes the pilus is referred to as the sex pilus because it helps in the exchange of genetic material. Okay, so by and large we have covered almost all the aspects of a prokaryotic cell. So remember, there are two different types of storage products. Some of them are bound by a membrane, some of them do not have a membrane, or they have a non-unit membrane which is about 2 to 4 nanometer thick in the case of gas vacuole, in the case of sulfur granules, in the case of polybeta hydroxybutyrate, where they are covered with a single layer of lipid molecules which have a hydrophilic head and a hydrophobic tail. It is very important for you to understand, for you to recall what is the function of these invaginations of the plasma membrane that are found, which is referred to as the mesosome. As opposed to what we just discussed, that is prokaryotic cell, you can see how distinct a eukaryotic cell looks like. It has a membrane bound nucleus with a region inside that is called nucleolus. There are a lot of pores in the nuclear membrane which are called the nuclear pores, correct? There is a plasma membrane. If it is a plant cell, it will have a cell wall on the outside. Otherwise, generally there is no cell wall. And you can see there are a lot of membrane bound cell organelles. For example, there is Golgi complex very close to the nucleus. There is endoplasmic reticulum. There is mitochondria. If it is a plant cell, it will have a chloroplast. There is lysosome, which are actually derived from the Golgi complex, which belongs to the endomembrane system. Sometimes the endoplasmic reticulum which have ribosomes on them are called rough endoplasmic reticulum. Those which do not have the ribosomes on their surface are called smooth endoplasmic reticulum. Centrioles are present, rod-like structures which are called as diplosomes, which are oriented at right angles to one another, and they are surrounded by an amorphous matrix that is called kinoplasm or the centroplasm, or it is also referred to as the pericentriolar material, which is referred to as the centriole. See how much difference is there in a typical eukaryotic cell in comparison to a prokaryotic cell. The smallest cell is mycoplasma, that is 0.3 micron meter. Then you have bacterial cells, which are 3 to 5. The single largest animal cell that has been identified is that of the ostrich egg. So please remember the isolated animal cell. Nerve cell is considered to be the longest in humans, and if you take a typical RBC for that matter, it has a size of about 7 micron meter in comparison to a bacterial cell, which is about 3 to 5 micron meter in size. Okay. Then, if you take a prokaryotic cell, again we are talking about a prokaryotic cell, it has three coverings. The inner covering is referred to as the plasma membrane, the middle covering is referred to as the cell wall, and notice this green covering on the outside, which is called as capsule. So, if I take a bacterial cell, this bacterial cell will have a cell membrane to begin with, then it will have a cell wall on the outside, and then it will have a membrane which is covering on the outside a capsule. Now, notice there are two types of capsule. If it is very loose like this, then it is referred to as a slime layer. Suppose you have, this is the cell membrane, the cell wall, and suppose if it is very rigid and very thick like what I am showing you here, then it is referred to as a capsule. So remember, the outermost layer, which is made up of polysaccharide and protein, if it is loose, the outer layer, if it is loose, then it is called slime. If it is very thick and if it is rigid and if it is very strong, then it is referred to as the capsule. Both the capsule and the slime are collectively referred to as the glycocalyx of the bacterial cell. It is the slime and the capsule that offer a kind of a sticky or a gummy nature to the cell wall, and they help the bacteria to adhere onto surfaces. And especially this glycocalyx also helps in escaping the immune system. Now, if there is a bacteria growing inside your body, it has the ability to escape the surveillance of your immune system with the help of this glycocalyx. And like I said, if it is loose, the glycocalyx is referred to as the slime. If it is rigid and thick, it is referred to as, if it is thick and tough and rigid, it is referred to as the capsule. So basically there are three coverings which you saw. The innermost covering is the plasma membrane, the middle covering is the cell wall, and the outermost covering is the glycocalyx, which may be slime or it may be a capsule. Now there is, again, depending upon the cell wall type, we can classify bacteria into gram positive and gram negative bacteria. Now notice the difference in the cell wall, all of you. Now this is the cell membrane, that is the plasma membrane of the cell. Now outside the plasma membrane you can see a very thick cell wall, and this thick cell wall, where you find, this is found in the case of gram positive bacteria. Gram positive bacteria has a very thick cell wall, and this cell wall is made up of a special substance that is referred to as murine or mucopeptide, or it is also referred to as peptidoglycan. Okay, so it has almost 70 to 80% of peptidoglycan in this surface layer, which is very thick in the case of gram positive bacteria. And you all can see a gap that is present between the plasma membrane and the cell wall. This gap is referred to as the periplasmic membrane, and along with murine, the cell wall also has a special chemical in it which is referred to as the teichoic acid. So this purple layer that you're seeing outside, the purple layer that you're seeing outside is made up of this peptidoglycan, which is referred to as the murine, and it has what is called teichoic acid, and the space between the plasma membrane and the peptidoglycan layer, as I mentioned, is referred to as the teichoic acid. But can you see how this middle, the cell wall layer, which has murine or peptidoglycan, is very thin? It has only about 10 to 20% of murine in it, and it has an additional wall. You can see an additional membrane is there outer to the cell wall. Now this purple layer is what we call the cell wall. So the inner layer is the plasma membrane, the middle layer is the cell wall, and outer to that there is an outer membrane as well. If there is an outer membrane, then we call it as a gram negative bacteria. Please remember, the outer membrane is made up of lipopolysaccharides. It is rich in lipopolysaccharides, and this outer membrane is a very unique feature of gram-negative bacteria. Now you can see two spaces. The space between the plasma membrane and the cell wall of peptidoglycan is called the inner periplasmic space, and the space between the peptidoglycan layer and the outer membrane is called the outer periplasmic space. So basically you can identify two spaces. One is called the inner periplasmic space and the outer periplasmic space. But most important important differences. See how this is very, very thick. The purple layer is very thick here. The purple layer is very thin in this case. And this purple layer, like I told you, is the cell wall made up of murine or peptidoglycan. Teichoic acid is absent in the cell wall of gram-negative bacteria. In gram-negative bacteria, there is a total of 3 membranes: inner membrane, middle cell wall, and outer membrane. The outer membrane is rich in lipopolysaccharide. The outer membrane is missing in the case of gram-positive bacteria. This was identified by using a staining technique called the Gram staining technique, which was identified or discovered or invented by Christian Gram. Gram-positive bacteria stain purple when you carry out the procedure of Gram staining, whereas the Gram-negative bacteria, they stain pink when you carry out the Gram-negative staining. So all of you remember this one thing. Cell wall is very thick. Cell wall has 70 to 80% murine. Cell wall is very thin. Cell wall has 10 to 20% murine. Teichoic acid is present in the cell wall. Teichoic acid is not present in the cell wall. Outer to the cell wall, there is an additional membrane called outer membrane rich in lipopolysaccharides. Okay. Ribosomes, as you all know, ribosomes are substances which do not have a membrane attached to them. They don't have an outer membrane to them. And you know that ribosomes have 2 subunits in them. If you take the ribosomes of prokaryotes, for that matter, they have a smaller 30s subunit and a larger 50s subunit. So if you imagine the larger subunit to be a structure below like this, the smaller subunit will fit onto it like a cap-like structure. So this larger subunit in the case of prokaryote is referred to as a 50s. In case of eukaryote, it is referred to as 30s. We call ribosome as nucleoprotein complexes. Now why we call them as nucleoprotein complexes is because, yes, any idea why it's called nucleoprotein complexes? Because it is a complex between protein and which nucleic acid? It is very, very rich in rRNA molecule, ribosomal RNA molecule with proteins in it, and therefore it is called a nucleoprotein complexes. Which are the types of ribosomal RNA that are present in the ribosome? If you say that there are what is referred to as, I told you that in this 50s, note that the values are not additive. 70s. Don't think 50 plus 30 is not 70. Then why it's called 70s? S is called Swedberg unit, which is a measure of its density, how quickly the particle settles once you centrifuge it. The values are not additive. So basically the 50s subunit has what is referred to as 34 different proteins in it. The 30s subunit has 21 different proteins in it. The 50s subunit contains in it 2 different types of ribosomal RNA, that is 5s rRNA, as well as the most important 23s rRNA is present in the larger subunit. The smaller subunit contains a small rRNA that is referred to as the 16s rRNA. So remember that the larger subunit of prokaryotes, which is a 50s subunit, has 34 proteins with 5s rRNA and 23s rRNA, whereas the 30s or the smaller subunit has 21 different proteins in it with 16s rRNA contained in it. Okay. Ribosomes are the principal sites of protein synthesis. Sometimes what happens is there is a thread-like messenger RNA which holds all the ribosomes together. See how the messenger RNA is holding all the ribosomes together, and this complex is referred to as a polyribosome, or simply it is referred to as a polysome. A polyribosome or a polysome is something but an mRNA which holds together. It's a string of ribosomes which are held together, and that means actually here proteins are getting synthesized. So these beaded structures represent proteins. So basically these are active sites of protein synthesis, and these are seen exclusively in case of prokaryotes. Inclusion bodies, sometimes there will be a lot of storage granules. I already spoke about this. Phosphate granules are there. Phosphate granules or Synophysian starch is there. Glycogen granules, they don't have a membrane. Sometimes you may find gas vacuoles and sulfur grains are present. Sulfur granules are present, and then I told you poly beta hydroxybutyrate crystals are there, which are covered by a non-unit membrane of 2 to 4 nanometer thickness. So we have discussed about the storage bodies which are referred to as inclusion bodies in bacteria. Cell membrane is present immediately below the cell wall in plant cells and bacterial cells, and it is present as the outermost boundary of the cell in case of animal cells. Cell membrane is usually covering the entire cell. It creates a distinction between the internal cellular atmosphere, that is the intracellular matrix that is found inside the cell, which is the cytoplasm, and the extracellular matrix that is found outside. In a eukaryotic cell, you have cell membrane in all forms of cell organelles like plastid, nucleus, mitochondria, and endoplasmic reticulum. There is also a vacuole that is seen in the case of plant cells, and the vacuole is also covered by a membrane that is referred to as the tonoplast. So you know about the fluid mosaic model that was proposed by Singer Nicholson in 1972. According to the fluid mosaic model, notice how there is a bilipid layer in the plasma membrane. Each lipid molecule has a hydrophilic head and a hydrophobic tail. The head is exposed to the outside and the tail is buried deep inside the plasma membrane. It is said that this plasma membrane has a quasi-fluid or a partly fluid nature, and all these purple colored bodies represent the proteins which are freely swimming in the protein. It's like icebergs. It's like how icebergs float in a sea water. These protein bubbles, which are basically represented in the form of purple structures, are freely floating over here, and this free movement is what is attributed to the fluid mosaic nature. That's why it is called fluid mosaic model, because the protein molecules which are suspended like the icebergs in the ocean can flow around freely. Some of the protein molecules are present superficially and some of them are present intrinsically. See, they are present all along the width of the plasma membrane. 70% of the proteins are present throughout the width of the plasma membrane. They are called intrinsic or integral proteins. Some of the proteins, it is not represented here or here. They have mentioned some of the proteins are present only on the surface, that is referred to as the peripheral protein, and these peripheral proteins make up 30% of all the proteins that are present in the plasma membrane. And you can see how some of the protein molecules are attached to carbohydrate molecule. When a carbohydrate is attached to a protein molecule, then we call it as a glycoprotein. Sometimes you can also find sugar units attached to the lipid molecules to form, suppose this is the lipid head, sugars will be attached to the lipid head, and then this becomes known as a glycolipid because it is a combination of sugar and a lipid molecule. Okay, see, these are all oligosaccharide chains. They are basically chains which contain about 2 to 10 monosaccharide units. And then it was identified that this fluid mosaic model was much more clear, as the structure of the plasma membrane was identified after the advent of electron microscope in the 1950s. And it has been identified that the cell membrane of RBCs, please remember, the cell membrane of RBCs are made up of 52% of the cell membrane is made up of protein. That means protein is such abundant in the cell membrane, and only 40% of it is made up of the lipid. And like I told you, the lipid is arranged in the form of a bimolecular layer which have these tails which are buried in the center and the heads which are protruding outside. These are called the polar or the hydrophilic head, and these tails are referred to as the non-polar or the hydrophobic tails. Cell wall, if you take a typical cell wall, basically the cell wall is found exclusively in plants. Of course, we also find it in fungal cell, in case of fungal cell, and I already discussed about the structure of bacterial cell wall. Gram-positive cell wall, which is very, very thick, and gram-negative, which is covered with an outer membrane. Gram-positive cell wall, which has 80% peptidoglycan, and only 10 to 20% of peptidoglycan is seen in the case of, in which one? In gram-negative forms. Now we are going to concentrate specifically on the cellulosic cell wall of plant cell. Now in the next picture, it will be very clear to you. In the lumen, you will find the plant, the actual cell of the plant, the plasma membrane with the nucleus in the periphery and with a vacuole in the center. Here it is called lumen, but I am showing you the cell now. So what do I call this cell which I am considering independent of the cell wall? I can call it as the protoplast. So protoplast has a membrane, it has a nucleus which is sitting in the corner and a vacuole. It has all sorts of cell organelles. Now let us see what a plant cell is covered by on the outside. The outermost covering is the red layer here, which is referred to as the primary cell wall. This primary cell wall is the outermost covering. After the primary cell wall is formed, several layers of cell walls are formed inner to the primary cell wall. Between the primary cell wall and the plasma membrane, notice this is the plasma membrane. So between the primary cell wall and the plasma membrane, several layers are laid down, and these layers of cell wall are referred to as the secondary cell wall layers. Notice that they are present inside the red layer. What is this red layer? The red layer was formed first. This first cell wall is referred to as the primary cell wall. Those layers which are deposited inner to the primary cell wall are referred to as the secondary cell wall. Both of them have cellulose in them. However, the secondary cell wall may have additional compounds like pectin and hemicellulose. And now there is another cell which is sitting beside like this. So let me take another cell over here. This is the primary cell wall, and I told you inside the primary cell wall there are 3 layers of secondary cell wall for the diagrammatic purposes, and inside there is the plasma membrane. See, inside the plasma membrane there is nucleus and there is vacuole, isn't it? So now you can see this is the primary cell wall of one bacteria, the red layer here, and this is the red layer of the next plant cell, not bacteria, I'm sorry, next plant cell. And now notice that there is a cementing substance that is present in blue over here, and this layer, which is made up of a cementing substance, is referred to as the middle lamella. It is made up of calcium and magnesium pectate. It helps in binding the adjacent plant cells together. So which color is the middle lamella? The blue color. What about the red layer? The red layer was formed first in the cell wall. It is called the primary layer. So in the diagram which I have written here, you can see inside the primary there is 1, there is 2, there is 3 layers. Which are these layers called? Secondary wall layers. Which is the innermost layer of the plant cell, the actual plasma membrane of the plant cell. And plasma, the cell alone without the cell wall, is referred to as the protoplast. And now you can see outer to the primary cell wall, you can see this blue layer which I have shown here in the form of a spiral, and this layer is called the middle lamella, which acts as a cementing substance between the two cells. Okay. The endomembrane system is a system of membrane-bound structures which are found inside the cell. The most important components of the endomembrane system are endoplasmic reticulum, the Golgi apparatus, the lysosome, and the vacuole. So remember the 4 major components that become the endomembrane system of a typical eukaryotic cell. Endoplasmic reticulum is present very close to the nucleus. See, this purple layer is the nucleus with openings which are called, actually it's the nuclear membrane with openings which are referred to as the nuclear pore. Yes or no? Immediately outer, you can see how the outer membrane of the nucleus, actually this nuclear envelope is a double layer like this, and this outer layer extends into to form flattened sac-like structures, and these flattened sac-like structures are referred to as the endoplasmic reticulum. So this is the endoplasmic reticulum. See how it continued from the outer wall of the nuclear envelope. Remember, the nuclear envelope has 2 layers, the inner membrane and the outer membrane of the nuclear envelope. The outer membrane is what continues as a system of flattened disc-like structure, which is referred to as the endoplasmic reticulum. See, you can see how it is system of flattened discs over here. These are the membrane bound discs which I am highlighting for you. Okay, now these membrane bound discs are referred to as the cisternae. In some places the cisternae are extended into hollow tubes which are referred to as the tubules. In some places they give out these vesicles, see, these sac-like structures which are referred to as the endoplasmic reticular vesicles. And one more thing is the endoplasmic reticulum here, you can see it is studded with these golden particles which represent the ribosomes. Whenever the flattened disc-like structure or the cisternae of the endoplasmic reticulum is studded with ribosomes, then we say that it is rough endoplasmic reticulum. And notice how the rough endoplasmic reticulum gives out a system of tubes or canals or even flattened membrane structures which do not have what? They do not have a ribosome on them. So that is referred to as the smooth endoplasmic reticulum. Now from the smooth endoplasmic reticulum, these vesicles come out, and these vesicles go on to join another cell organelle that we will be discussing now, that is called the Golgi complex. So remember that the smooth endoplasmic reticulum is a bridge between the endoplasmic reticulum and the Golgi apparatus. Please go through the functions of endoplasmic reticulum. All the protein synthesis, obviously because it is attached to ribosomes, protein synthesis, processing, packaging of proteins, everything happens over here. The packaged proteins are transported by these vesicles, isn't it? and these vesicles are the ones which usually provide the raw material to the next cell organelle in line with very close to the endoplasmic reticulum, that is referred to as the Golgi complex. Endoplasmic reticulum, especially the smooth endoplasmic reticulum, is involved in detoxification of drugs in our liver. It is involved in steroid and lipid synthesis, and it also acts as a bridge between endoplasmic reticulum and Golgi complex. Another important thing is the rough endoplasmic reticulum, which has ribosomes in it, is the site of glycosylation. So all of you remember that RER is a site of rough endoplasmic reticulum, is a site of glycosylation. Glycosylation means addition of glucose or carbohydrates to proteins, that is referred to as glycoprotein synthesis. So whenever you add carbohydrate to a protein, it becomes a compound called glycoprotein. The process of adding a carbohydrate to the protein is mostly carried out by this particular endoplasmic reticulum. So remember, these are sites of glycosylation. Okay, so endoplasmic reticulum, you have to remember that it is mostly involved in I'm so sorry, the sites of glycosylation is not rough endoplasmic reticulum, it is Golgi complex. In the next slide I will discuss about this in detail. For now, forget this point. Like I said, the rough endoplasmic reticulum has ribosomes on it, so it is obviously helping in protein synthesis, and then I told you it helps in packaging all the proteins, and it also helps in what is referred to as especially the smooth endoplasmic reticulum. I want all of you to remember the smooth endoplasmic reticulum helps in detoxification, and I'm writing it here, it helps in lipid and sterol synthesis. So ignore the glycosylation part, I will talk about it when I discuss Golgi apparatus. So lipid and sterol biosynthesis, and then I told you that they help in detoxification of drugs. Another surprising function of smooth endoplasmic reticulum is it is a storehouse of calcium, especially in muscle cell, and it also passes on, like I told you, it passes on these secretory substances to the Golgi complex. So let us see how a Golgi apparatus looks like. See how from the endoplasmic reticulum you have these vesicles which are carrying substances, and they are coming and merging with this face of the Golgi apparatus. Obviously somewhere here you have the nucleus of the cell. I told you the endoplasmic reticulum is a continuation of the outer nuclear membrane. This face of the Golgi complex, which is basically facing the nucleus, is referred to as the cis face, and the face of the Golgi complex that is facing away from the nucleus and the ER is referred to as the trans face. So remember, the Golgi apparatus is very closely situated to the endoplasmic reticulum. So actually what you should remember here is basically it is not directly from the rough endoplasmic reticulum. Like I told you, from these the vesicles enter into the smooth endoplasmic reticulum, and these smooth endoplasmic reticulum, which are nothing but in the form of tube-like structures, branch tube-like structures which don't have ribosomes on them, they are the ones which carry these vesicles into the Golgi apparatus. See, in the Golgi apparatus what happens is whatever proteins were synthesized in the endoplasmic reticulum, they undergo processing, they undergo packaging, and like I told you, this is the site of glycosylation. A protein is added with carbohydrate to become a glycoprotein, a lipid is added with a carbohydrate to become a glycolipid. So basically Golgi apparatus is nothing but a site of glycosylation, is what you should remember. And see how vesicles are coming out of the Golgi complex, and these vesicles are going and attaching, or they are going and reaching their destination, which may be the plasma membrane of the cell, the outermost covering of the cell, which is the plasma membrane. Okay, so whenever you think of Golgi complex, please remember it is involved in processing, packaging, transport of material coming in from rough endoplasmic reticulum through the smooth endoplasmic reticulum. And I told you it is the site of glycosylation, where carbohydrates are added to protein to form glycoproteins, and carbohydrates are added to lipids to form glycolipids. And we also saw that this Golgi complex is also found in the acrosome of the sperm. If you remember, the acrosome is a vesicle sitting on a cap-like structure sitting on the nucleus of the sperm. This is actually derived from the Golgi complex, and the Golgi apparatus also helps in cell plate formation in plant cells. It also helps in cytokinesis of both animals and plant cells, isn't it? Division of the cytoplasm in plants, specifically by the formation of cell plate, which divides the cell into two daughter cells, and very importantly you can see a lot of Golgi complex in the root cap cells, and in the root cap cells the Golgi complex secrete a slimy sticky substance that is referred to as the mucilage to lubricate the root cap, because the root is continuously burying itself into the ground, it is growing into the ground, it may get damaged by the soil particles or the sand particles or the grits in the earth, and as a result the root cap is protected by a mucilage which is secreted by the cells of the root cap, which are very rich in Golgi apparatus. Okay. Lysosomes are originating from the Golgi complex, and these lysosomes are nothing but small sac-like structures. These are vesicles which are bound by a membrane, and remember they are formed by a process of Golgi apparatus, and inside the lysosome they contain these very important digestive enzymes which is referred to as the hydrolytic enzymes, which help in digestion of or breakdown of lipids, breakdown of proteases, breakdown of carbohydrates. They were first reported by Christian D. Dewey in the year 1955. Vacuoles are membrane bound. 90% of the plant cell is occupied by vacuole, and vacuole is covered by a unit membrane that is referred to as a tonoplast. And then remember that the vacuole contains water, it contains salts or sap and excretory substances, that is mostly which is not required by the cell. And more importantly, vacuole is very high in ionic concentration because the ions are pumped into the vacuole from the cytoplasm against the concentration gradients. Obviously all the ions get pooled inside the vacuole. So vacuole is rich in water, sap, that is referred to as the sap vacuole because sap is basically a mixture of salts, okay, excretory substances, and a lot of ions are present inside the vacuole which are actively pumped from the cytoplasm by consuming energy. Mitochondria. Now you're all familiar with mitochondria. I'm not getting into the details of mitochondria because you can manage it on your own. There is an outer membrane that is it is a sausage shaped body. It doesn't mean that all mitochondria are sausage shaped. They may occur in varied shapes and sizes. There is outer membrane, and then you can see the inner membrane, isn't it? Now the outer membrane has about 60% protein and 40% lipid, but the inner membrane has about 80% protein and 20% lipid, and the inner membrane of mitochondria is made up of a special protein or a special substance which is called the cardiolipin. And like I said, the inner membrane has 80% protein and 20% lipid. So most of the inner membrane is protein and only 20% of the inner membrane is lipid, whereas the outer membrane, it is 60% of the outer membrane is protein and 40% of the outer membrane is lipid. And notice how between the two membranes you can find a gap. This is called as the perimitochondrial space. The gap between the outer and the inner mitochondrial membrane is called the perimitochondrial membrane. And notice how the inner membrane is thrown into finger-like projections or finger-like outgrowths which are growing into the matrix. The matrix is referred to as, it is simply called the matrix, the fluid substance that is present inside. And remember that these finger-like structures grow into the matrix and they contain special enzyme complexes. You can see this enzyme complexes here which has a basal disc and a stalk and a head, which is referred to as the F0, F1 particle or Racker's particle or oxysomes, and these are the ones in which ATP synthesis takes place during aerobic respiration. You all are aware. So it is usually also referred to as the ATP synthase. All right. And then there is a double-stranded naked DNA which is referred to as the mitochondrial DNA, and the fluid inside the mitochondria is referred to as the matrix. You know that the mitochondria is the site of aerobic respiration and it provides a lot of energy to the cell. Plastids, in which we are focusing about the chloroplast. Just like mitochondria, a plastid also has an outer membrane and an inner membrane. The fluid in this case is not called matrix, we refer to it as the stroma. There are several membranous disc-like structures which are stacked one above the other. Each membranous disc-like structure is called a thylakoid. The entire stack is referred to as the granum. Many stacks are collectively called grana, but since I'm showing only one stack, it is referred to as the granum. And you can see how one granum is connected to the neighboring granum with the help of these membranes, which are referred to as the stromal lamellae, or what is referred to as the fret membranes. Okay, even inside the chloroplast you will find lot of ribosomes attached in a beaded structure on mRNA. We had discussed the same in bacteria. They were called as polysomes, and they also have a double-stranded DNA, a naked DNA, that is referred to as the chloroplast DNA or the ctDNA. You all are aware that in the granum the light reaction of photosynthesis takes place in the presence of sunlight, and in the stroma the dark reaction or the C3 pathway of the photosynthesis takes place. It is called dark reaction not because it happens in the dark, because it does not require light to occur. Okay, chromoplasts are yellow orange, just like chloroplasts. They don't have grana in them. They are yellow orange or reddish pigments containing vacuoles, sorry, not vacuoles, they are plastids, which are yellowish or orange or reddish, and they appear colored because they contain carotenoid pigments. The color of carrot, which is the orange color, is because of the presence of these plastids, which have these yellow, orange and red kind of colors or pigments in them. And then you can see how the change in the color of the tomato and chili when it is raw and when it starts becoming ripened, that is because there is a transformation of chloroplast to chromoplast. A chromoplast can come from both a chloroplast and what is referred to as a leucoplast. A chloroplast can transform, it can lose its grana, it can change its chlorophyll molecule in order to become a chromoplast. Like we said, as the fruit starts maturing, the color, that is the green color inside the chromoplast, is getting replaced into the reddish or the greenish to reddish coloration. So basically what transforms into the chromoplast, either a chloroplast can transform into a chromoplast, or a leucoplast can transform into a chromoplast. Leucoplasts are entirely colorless. At least we saw chloroplast was green in color, chromoplast was occurring in yellow, orange and reddish colors. But there are certain plastids which are present in the plant cells which have absolutely no color. They are called leucoplasts. They are meant for storage. For example, those leucoplasts which store starch, that is amylose, they are referred to as amyloplasts. Those which store oil are called eleoplasts or oleosomes or lipoplasts, and those which store proteins are referred to as aleuroplasts. So whenever you revise about leucoplasts, you have to remember the three types of leucoplasts which are basically present inside a typical cell. That is basically you have amyloplasts, which are meant for storage of carbohydrates, eleoplasts or leucoeleoplasts, or lipoplasts or oleosomes, which are meant for the storage of fats, and lastly you have aleuroplasts, which help in the storage of protein. Ribosomes I have already discussed with you. The only thing I wanted to tell you here is it was discovered by George Palade in the year 1953. As a result, ribosomes are referred to as the Palade particles. I told you in case of prokaryotes it is 50 and 30 subunit, that is the smaller one is a 30s and the larger one is a 50s, and totally it is a 70s in the case of prokaryote. In eukaryotes, the smaller one is a 40s, the larger one is a 60s, and collectively it is referred to as the 80s type in the case of eukaryotes. I had also told you that the 30s subunit has 21 different proteins, 50s subunit has 34 different types of proteins, 40s subunit has 21 different types of proteins that we saw, and then the 60s subunit has about 40 different types of proteins that we had discussed, isn't it? So basically you have to remember these proteins, and I had also told you about these being nucleoprotein complexes. They have protein along with rRNA, which is the rRNA found in the 30s subunit of smaller subunit of 70s particle. It is 16s, very good. Which is the rRNA found in this case? It is 5 and 23s. In this case it is 5, 5.8 and 28s. In this case it is 18s. So you should remember how many proteins are there and how many types of rRNAs are there inside the ribosome. Cytoskeleton is of two types. Basically there are microtubules which are cylindrical, they are hollow, and their wall itself has individual units which are referred to as protofilaments, and you have microfilaments which are nothing but they have a very narrow diameter, they are usually actin, and you have intermediate filaments which are comparatively thicker in comparison to microfilaments, but they are thinner in comparison to microtubules. Now if we were to look at the structure of the cilia and flagella, this is the cilia and flagella. Their structure is almost the same, but cilia is very thin in comparison to the flagella, and cilia are small hair-like structures, where flagella is a whip-like structure. Here we have taken a section of the flagella. Notice that there is a central very complicated structure that is referred to as, this is referred to as the axial filament. So the axial filament or the axoneme, notice that there are doublets of microtubules which are present. I told you a microtubule is nothing but a hollow tube like this, and in the wall there are 13 tubes. I'm not counting it, I'm just drawing roughly, and each tube has a repeated unit of alpha beta protein dimers, and one such cylinder or one such tube made up of these repeating units are referred to as the protofilament. And a microtubule is composed of how many protofilaments? 13 such protofilaments. It is mostly 25 nanometer in diameter, and the hollow core is mostly about 15 nanometer in diameter. So basically, how many such if you look at a protofilament, it will have alternating alpha beta tubulin, alpha tubulin, beta tubulin, alpha tubulin, beta tubulin. So this whole thing is referred to as a protofilament. How many such protofilaments are present in the microtubule? 13 different protofilaments are present. See, so 13 protofilaments are arranged around a hollow center in order to give rise to this hollow tube, which is referred to as the microtubule. So what you see in the axial filaments are basically microtubule. You see doublets of microtubule, that is one complete microtubule which has 13 protofilaments, and outer to that you will find an incomplete protofilament which had about 10 to 11, and these are referred to as the microtubule doublet. So remember that cilia and flagella have a microtubule doublet. How many are there? Let us count. 1 2 3 4 5 6 7 8 9 10. Sorry, 9. So 9 microtubule doublets, and in the center you can see 2 are microtubules which are single, and they are covered by a sheath which is called central sheath. And can you see these spoke like structures which are extending? These are also proteinaceous. These extend from the peripheral doublets to the central sheath, and they are referred to as the radial spokes. So this is the outline of the structure of cilia and flagella. Centrioles and centrosome. In a centrosome, you have two right-angled oriented structures, rod-like structures, which are referred to as the diplosome. And you can see that there is a matrix of amorphous substance that is present outside this particular diploplasm, and that matrix is referred to as the centrosome. Pericentriolar material. It is called PCM or kinoplasm or centriolar matrix, you can say. That is pericentriolar material, and this pericentriolar material does not have a membrane attached to it. And notice, if you take a transverse section of this centriole, you can see that they have microtubular triplets. Each structure here at the periphery is a set of 3 microtubules. That is, one is called A microtubule, which is a complete microtubule, then there is B, which is an incomplete microtubule, and then there is C, which is an incomplete. So imagine there is one triplet here and there is one more triplet next to it. Generally the C of the microtubule triplet and the A tube of the microtubule triplet are connected together by proteinaceous substances which are referred to as the linker. So this is A, this is B, this is C. Is it clear? So this gives the entire centriole the structure of being a cartwheel in appearance. And notice in the center there is no microtubule singlet. There is a mass of protein in the form of a rod-like structure that is called as hub, and you can see these protein threads or protein strands which extend from the hub to which is this complete microtubule at the peripheral triplet. I told you the complete microtubule is A, and the remaining ones are referred to as B and C, which have about 10 to 11 protein filaments in them. And basically what you need to know is that there are proteinaceous strands which extend between the hub in the center to the peripheral microtubule triplets, and these are referred to as the radial spokes. Okay, sometimes around this particular centromeres, around the centriole or the centrosome, you can find certain bodies, certain aggregations. These are referred to as the satellite bodies or pericentriolar substance. Okay, and these basically help in organization of the spindle fibers, so that we call them as motor spindle assembling points, which is also referred to as the microtubule organizing centers. Now these structures, these satellites, which are called pericentriolar satellites. These are aggregations which are present around the centrosome, and these pericentriole satellites is where microtubules are formed. So they are called microtubule origin centers, or they are called microtubule organizing centers, that is MTOC, microtubule organizing center, and these are also referred to as the pericentriolar satellites. Is that clear? Nucleus, it has a nuclear membrane. Like I said, there is a double membrane. There is an inner nuclear membrane and outer nuclear membrane, which extends as the nuclear envelope. In between the two membranes, there is a periplasmic perinuclear space, and then you have the nucleoplasm, which is the substance within the nucleus is the nucleoplasm, and non-membrane bound collection of chromatin or DNA material, which is called nucleolus. Nucleolus is the site of rRNA synthesis. You have to remember this very importantly. Then there is nucleoplasm. There are nucleopores. Nucleopores are those regions where nuclear envelope is not present. Substances are allowed to move in and move out, of course, with strict surveillance. Okay. Then you have the nuclear. Yeah, we already finished about the nuclear membrane. And notice how the outer membrane of the nucleus is in continuation with endoplasmic reticulum. Chromosome classification based on centromere. Exactly in the centromere you have metacentric, slightly away from the center predisposed submetacentric, closer to the periphery, closer to the polar region but not entirely, that is further away from the center acrocentric. Exactly at the end of the chromatids it is referred to as telocentric. So please learn this from the textbook thoroughly, the modifications or the variations in the height of the variations in the position of the centromere when it comes to the chromosomal structure. Okay. Microbodies are membrane-bound substances. These microbodies are found both in plant and animals. Let us see some of the microbodies. Peroxisomes are very common in plants. They are very closely associated with mitochondria and chloroplasts. They have enzymes that are required for the synthesis of peroxide, that is hydrogen peroxide, that is glycolate oxidase. That is basically the one enzyme oxidase which helps in synthesis of most importantly you have what did I say? Peroxide. There are enzymes which degrade peroxide within the peroxisomes. The enzyme which degrades peroxides is called catalase. So two types of enzymes. Glycolate oxidase is an enzyme that synthesizes peroxide, and catalase is an enzyme that breaks down peroxide to obtain water and oxygen. If hydrogen peroxide is synthesized beyond a point and it is stored in the cell, then it can be harmful to the cell. As a result, it is broken down by catalase enzyme. Remember, peroxisomes are very common in both plant and animal cells, and mostly in plant cells, and peroxisomes also take part in a very important pathway in photosynthesis that is referred to as photorespiration. Okay, so you will learn this in the further, you have already learned it in photosynthesis chapter. Glyoxysomes are exclusively found in germinating seeds, and these glyoxysomes are microbodies which are small vesicles covered by unit membrane. They take part in a cycle that is referred to as the glyoxylate cycle, through which fats are converted. Now fats undergo a process called beta oxidation of fats, and in that you have acetyl coenzyme A, that is a byproduct. So a byproduct of fat breakdown is referred to as acetyl coenzyme A, and this acetyl coenzyme A, through a process called glyoxylate cycle, will help in the synthesis of glucose. Now since glucose is synthesized from not a carbohydrate, we call this process as gluconeogenesis. Neo means new. You are synthesizing something from something new. So glucose is being synthesized not from another carbohydrate but from acetyl coenzyme A. Acetyl coenzyme A is a byproduct of beta oxidation of fatty acids, and this acetyl coenzyme A, through a cycle called glycolate cycle, will generate what is referred to as the glucose, and that is the major function of glyoxysomes. So with this I complete the very quick recap or the synopsis of the chapter Cell, the Unit of Life. Thank you.